So we're talking about soil health tests and how should we use them. This is a topic of, I would say, fairly great interest over the past 5 to maybe 10 years ago. I did a little bit of research here, and it looks like the concept of soil health testing was birthed around 2006 in that era. So we're talking over 10 years ago. And I know for myself, when I first heard about them, I thought this is just exactly what I mean. This is what we need. This is going to really prove what cover crops can do for the soil. And since that time, there have been numerous soil health tests that have hit the market and are out there and available. I'm going to run through some of them. And then I've done over 1,000 of them here in my farm just to try to get a handle on what they can and cannot do. So I'm just going to say in the outset here that there are some people who are very excited and very confident in what they can do, but there's also some people who are very antagonistic and basically say they're not calibrated and you can't depend on them. And it comes from a wide sector of the agriculture community in that. So there definitely is not a unified perspective on the value of them. And that's partly why I wanted to do this topic, because I definitely have a perspective, and I just want to share it with you as a way to help you and also collectively help us all be able to use these for the best aspects that we can. I also want to say on the outset that I'm going to share some of the concerns I have and some of the things that you probably cannot expect them to do, but I'll put myself in one of the biggest cheerleaders to make them be more reliable and more consistent, because I think that's what we definitely need in the future moving on, because they can really be helpful in eventually helping in our management, particularly with fertility. So I was first introduced to the soil health test by Jill Clapperton, who was at my farm in 2011. We had a field day, our annual field day at that time, and she brought these little strips that she collected soil. She was there the day before the field day, collected soil, put the strip in the soil, and then the next day looked at the strip and compared it to a comparison that kind of indicated a color change. And so that was essentially the precursor to the Solvita test, which probably most of you heard of the Solvita test. That basically measures the CO2 release over a 24-hour period from the soil. And then that is used then to just give a baseline of what the microbial activity is and so forth in the soil at that time. So I have been familiar with these tests and for the last couple of years, and certainly have tried them and seen how that they work on our farm. So that's a little bit of the history for me personally. So I'm going to go through some of the more popular ones here, and I already mentioned the Solvita. It's sometimes they call it the Solvita one-day CO2, which is basically a measurement of probably one of the more important numbers in the context of soil health reports. So this is a parts per million number that come out of this, and it's basically the amount of CO2 released in the 24-hour period from the soil microbes. So it's based on microbes, microbe activity. And this is through a process that the soil is dried, then re-wetted, and then that's just an indication of microbial activity. So the reason this is important, because there is a relationship to the amount of CO2 released, can be somewhat of an indicator as to what the fertility could be in the soil at any given time. So that's a Solvita test, primarily developed by Woods End Lab up in Maine. They have a man by the name of Will Britton, who's fairly well known in the soil health community. He's been working at this for decades, and I would have to say I've read some of his literature back in the 80s, and I would say he was a man ahead of his time as a researcher, and now some of his work is finally bearing fruit. So that's a little background on the Sovita test. Most of us have heard of the Haney test, which is Rick Haney is a USDA researcher based in Texas, and this has been his passion over the last 10 or 15 years of trying to understand soil health and so forth. So he developed a test that an integral part of his test is the Sovita test. So use that as a basis there. But he takes it a step further and measures some other parameters. He uses a water extractable organic carbon number he gets out of that. And the reason it's important to know about the water extractable is it's supposed to more like mimic what a plant will actually see or what a plant can actually experience in a real world situation. That's the whole idea behind it. So instead of using chemistry to figure these things out like has been done in the past, they're using water to look at organic carbon and organic nitrogen and so forth. But he also adds in the NPK chemical analysis component just to use that as kind of a background. And then what he does in his Haney test, what's become to be known as the Haney test, is take those different parameters there, and there's a calculation and a formula that equals a soil health score. So you send a sample in, they run it through these different things here, and they come up with a soil health score. And it's basically on a scale of 1 to 50, and basically a number over 7 is starting to get fairly good. If it's below 7, it's pretty poor quality soil from a soil health standpoint. So I will say that the Sovita component is weighted heavily in this formula. So the microbial presence in the soil is definitely one of the primary factors in understanding his test. Another one is the Cornell soil test, and this has probably been out in the public a little bit sooner, 2006, I believe, is when it was released. And the Cornell soil health test was from Cornell University in New York, and they look at a lot of different parameters, and I've listed them here. You can read them as I talk, but they look at it's more, I'm going to say, more complex. But then again, you could argue you get a little bit more information out of it. So they have this all well outlined and what to do. Primarily researchers have been using this. It is cumbersome for farmers. Farmers just don't typically have the time, the effort needed to get all these different parameters. Some farmers do. If you're really interested in knowing this, there's some farmers do it, but I would say that the Cornell one is more comprehensive, but then again, it takes a little more time and effort to get the resulting numbers. And there's also add-ins there. There's like 4 or 5 other things that you can get tested and add it to it. And so that's just one thing that one of the more popular tests. So Dan Towery just asked what the cost is. I'll just back up. For the Haney test, it was in the $50 to $70 a test range. I know there is an attempt to get some cost share through the NRCS for it. I do not know where that's at right now. For a while there, he was doing anyone associated with research. He was doing them for free, basically just to kind of get up to speed with get the word out, so to speak, but also to try to better understand it and its predictability and all. The Cornell soil health test, I do not know the cost of that one, so I don't know the answer to that. Now there's another one here. It's a little less popular. Come on a little bit later. It's called the phospholipid fatty acids, or PLFA. I'm used to just saying PLFA, so I had to hesitate, look at that front word there. So the PLFA test is, it seems to be promoted mostly by Ward Laboratories out in Nebraska. Ray Ward and his lab has been, I would call them a leading lab in the country that gets it, the soil health component. They've been, I know that they have been, giving recommendations specifically for no-till farmers and cover crop farmers for years now, and they have a whole array of soil tests that they will have. They don't have the Cornell one, to my knowledge, but they do have the Haney, the Solvita, the PLFA. So basically, the PLFA has to do more specifically with the critters in the soil, the microbiological component, all the bacterial groups, Actinomyces, Arbuscular, mycorrhizal fungi, Rhizobia, protozoa, and all the microbiology within the soil. So but you gotta understand here that it's a snapshot. That's a key word here. It's a snapshot of microorganism community structure and abundance at the time of sampling. So that's key, especially with all these tests I've mentioned have a strong biological component. Just to summarize here with the PLFA is not so much to make fertility recommendations. It's more to compare management systems for farmers who would like the comfort of knowing that they use cover crops for a couple years versus a field that isn't. Is there a difference? It should show up in a test like this. So as far as using the PLFA for specific management decisions, I would say at this point it's not going to be a huge component in that. So I'll just wrap up my talk about the different ones here by saying that there are other tests that I'm beginning to hear about, and I did not list them because some of them I'm not sure how far along they are yet or not, if they're have the merit of being listed, but most of them are new. They take a different angle, or they're variants of improvements of what's already out there. So it's something that is definitely increasing, and I'm really happy to see this. But I would say we're not going to get real serious tools here anytime soon as far as specific recommendations when it comes to fertility inputs. In other words, we all would agree that our basic soil tests that we have used, number one, I'll give some perspective. Our basic soil tests that we have used haven't really changed much over the last 50 or 60 years. So, there's definitely room for improvement there. But that being said, I think we'd all agree that those soil tests are not an exact science, but they are a good indicator. So I would put the way I would rank our soil tests, I'm going to call them the traditional soil tests that measure phosphorus, potassium, calcium, and some of the micros. I would put them at their value in the context of a 3-day weather forecast, okay? So in 3 days, forecasting is pretty decent. You generally know what's going to happen in 3 days. You can manage for the 3-day weather forecast, and we do it all the time. Does it always work out? No, but it does most of the time. And if you want to put another layer of perception here, I would say 80-85% of the time a 3-day weather forecast, or 80%, 85% of the time our typical cover crop, or typical analysis for soil tests, chemical analysis, about 80-85% of the time you could probably say that they're consistent. Now when I wrap up here, I'm going to tell you what my perspective is on these soil health tests and where they're at currently. So I'll leave you hanging there a little bit. In the meantime, I'm going to share some of my own data that we took on our farm. And obviously, I can't show you, I believe it was almost 1,500 tests that we did over a couple year period. And I'll just say that most all of those tests were the Haney test. So I'll just say that, and if, in case I forget it, I am going to be doing the Cornell test this year, working with another project from another group. But so what you're seeing here is with Haney test. So I have the title of these next slides titled Managing What You Measure. And ultimately, that is the whole point here, to be able to manage what we measure. So are these measurements good enough to influence our management? That's what it's all coming to be. So I'm going to show you real data. This is actual data, including all the replication. So I just wanted to show you this here. This could be, for those of you who understand research and agriculture, this could be published data here. It's a randomized block design. And again, I'm not going to go over it all. You can see the treatments on the one side. That's not the point here. I just want to show you that this is what the data was. Now, this was corn. So corn was the crop we were looking at, and we were looking at several different cover crops with a control and with different mixes of cover crop species. So each block, the length was 100 feet, and the measurements were taken of 2 rows of corn. So that equates to about 1/100th of an acre. So yeah, I know that's fairly small, but when you're doing hundreds of these plots, I don't have a big enough farm to make my plots a lot bigger. So keep this all in mind, that it is relatively small, but it's not like a 10-foot plot either. So 100 feet by 2 rows. So just to give you that understanding of what is behind these numbers that I'm going to share with you then. So just to show you what the end result is here, and I'm not going to go over this. I just want to show you this is the kind of numbers we got out of that. This is just like a snapshot of like 1/10 of it or less. So there's a whole lot of data there, and there's a lot of stuff there. I'm not going to go into that. But boy, is it ever fascinating when you can look at numbers like this, especially if this is what really you're interested in. So I just want to show you the breadth of what we did here in looking into if we could use soil health tests on our farm. So now to some of the actual results, and you're gonna have to stick with me here because I can't use all the animations that I want to as I would live, because the internet's too slow and I don't want to have little things jerking around. But I am going to, I'm not going to use my mouse because it's too slow. So let's look over at the far left-hand side. You might wonder what the word Indy is. That happens to be the name of a mix of annual ryegrass, crimson clover, and radish. Okay, so that's a mix. That's a brand name of a mix. So that was the cover crop here. Then you look at the next line in, that's the nitrogen rate that was applied. Toward the bottom set is 3 of plots of 0 nitrogen. The 75 pounds had 75 pounds at planting, 2 by 2. The 150 had 75 at planting, and then 75 broadcast dry, combination of urea and protected urea and ammonium sulfate. So that's a little bit of background in that. And then you see the plot IDs next. They're scattered all over the place, so good data. So we took tests in these fields 3 times. The spring test was immediately after planting, before crop emergence. The summer test was right at side dress time, would have been late June, early July. And the fall was after harvest. So backing up here a little bit, this number that you see in here under the spring, Solvita, I kind of kept my column smaller there, so it's the Solvita 1-day CO2 burst. You see that number there on top, it says 50.03. That's parts per million of the CO2 measurement. The higher the number, the better it is. Or, as this is color-coded, green color or dark green is good, red is low. So that you can get a quick glance there. So I want to go over a few things here that I think are interesting to point out. So let's look at spring. Solvita on the 150 rates. There's three of them, three different plots. Number one is 50, the second one's 26, and the other one's 29. Now, when I look at that from a researcher standpoint, that is a little bit that's not as consistent as I like to see. So that means that we didn't find consistency in our measurements there. Now, of course, when you're doing research, the consistency is important. So I just want to make note of that. Look at the next set down of 75 pounds. This is 39.79, and the other two below it. We see that bottom one there, and that rate is a 45. And then you look at the zeros, that's a little lower. Now, let's just think back a little bit bigger picture here. Essentially, all those 9 numbers under spring were exactly the same because it was right at planting. So they essentially at that moment had 0 nitrogen. My concern with this is you see a low of 24.2 and a high of 50.03. That's highly variable coming out of the exact same field. Same field history and everything. There's no background legacy stuff here that we're aware of that would cause this. So that's a little not what I was hoping for. With sending these tests in, of course, they're sent in blind or sent in coded. So I'm just putting it out there. This is real numbers that we saw. Then when we took the data in the summer and the fall, even though in the fall it did narrow up, if you would look at that a little bit, after there was some fertilizer applied, half rate and full rate, it did narrow up. But let's look now over to the extreme right on the yields. So the top 3 there is the 150 pounds of nitrogen. When I see numbers like that, 195, 185, 199, that's good data. I really like to see that because it's consistent. In the context of what the measurements is here, I like that. That's good. You come down to the next set, the next 3, spreads out a little bit. And then you get to the bottom, and of course there was 0 nitrogen applied, so you can see there's not, we didn't get the yield there. And those numbers are a little bit wide there for whatever reason, but they are. So I just want to say that admittedly, I was a little disappointed with some of the lack of consistency, but when we put essentially a decent rate of nitrogen. I mean, I could have put more on, but I didn't want to over apply here. We got good yields when we put on the nitrogen as we would expect. So I feel like that kind of verified what we would expect. So now I want to move on to the next slide here that shows you, again, this is simply additional data of the very same research. So let's look at, again, the variability was a low of 9 and a high of 52. And again, when this was done in the spring, this was essentially 0 nitrogen was applied. So why do we have that disparity there? Again, look over at the top right with the yields of this is triticale planted late here. The yields were very, very close. That's good data. I love that data. It's very close. When you see the reps close, that means your probability of repeatability is much higher. So, and this is something that I'm just using this comparison here. It's something that I would expect when you put a decent amount of nitrogen on, you're going to get consistent yields. And I'm trying to use this as a basis for saying I don't understand why we had the variability with our tests here. So, so one thing that is interesting, though, I will say, is look at the 9.74 that's circled, and then look at the yield. That was the lowest in that plot, and sure enough, it yielded the lowest. Which there is a correlation here. I'm implying this. I've been implying this, but there's a correlation to the amount of CO2 burst and the amount of nitrogen needed to grow corn. So there, that was a nice, I guess you would say, a nice result in that. But when we look at another one down here, let's go down to the triticale late 0 and go into the spring. We had a high rating there, 47, but it really tanked on us. It was the lowest yield of those set of plots. So had I used 47 as a management tool, I probably would have only applied 75 pounds of nitrogen. So in this specific plot, it didn't work out. It yielded a lot less. So I'm just, again, showing you real data here that I found. And then finally here on the very bottom, the lowest testing plot was 15 parts per million, but that yielded almost equal to the highest in those plots with no nitrogen. I don't like that inconsistency with that. So I'm just going to say that in all the testing I did, I found that the test was accurate about 60% of the time. And that's with, like I said, over 1,000 different reps, or 1,000 different tests. So that to me was interesting, not what I had hoped for. Now, I need to mention this was 2013, and since 2013, I would like to say they have gotten better, but I haven't been consistently finding research by others that have said they have been a lot better. I hear anecdotal evidence that they are better from some people that are indeed using them, and they are getting good yields. We probably all heard the stories out there, but I've heard just as many stories who says it has not come through. Now, I'm not saying that we shouldn't use soil health tests because I think they are our future. I just want to make sure we understand here that we don't use them right away exclusively for our nutrient management decisions on applying extra fertility and so forth. So, how should we use soil health tests? Here's a few things. I think if you're interested in this, to use one of the ones I mentioned or new one that's coming out there, use them as a baseline. Don't test all 40 of your fields just to see where you're at, unless you have money to blow. But use them as a baseline. Where are you at? Just to get familiar with them. And also to compare management practices. One of the things that convinced me to try the soil health test, because I wasn't the first one to try them, is I had heard someone who was a crop consultant, and they said that they could pick out every field that had manure applied. It was fairly consistent with that. And so I thought, well, that's good. That's a good sign. So this is just one thing that you can use the soil health test for. But I will say that you really want to start small if you're using soil health tests to manage for fertility. Because how do your soils respond? And I emphasize there your soils, either on your farm or the farms that you consult for. How do they respond to these to fertility? There is so many variables involved in this, and the soil health tests are decent. Don't get me wrong, but to be able to account for all those variables is one thing that is tough to do. Now, I should have mentioned earlier, we're dealing with biology here, okay? So one of the things that can happen is people don't take care of their soil health tests. When they take their tests, if you would subject that test into the hot sun, you could kill a lot of things that are in there that could impact the results of that test. So we were told to put the test in a cooler immediately. And we did that. So, even though you see these numbers here that I showed you, we drug a cooler around in the field and packed it full of all these tests so that immediately after they were pulled, they were cooled, they were in the dark, and they were sent off in a package that was insulated so it wouldn't get extremely hot, because this is in the summer. That's what we were concerned about. So, So it's just something to be aware of. So I used my analogy previously that I feel that our current traditional soil testing is about as good as a 3-day weather forecast. I'm saying currently these soil health tests are about like a 7-day weather forecast. So it shows you what's coming up. It gives you a general direction, but not consistent enough to make major nutrient management decisions. So the other thing too in this aspect here is measuring your soil's health over time, because I firmly believe that these tests are going to get better and more consistent. So I want to be careful that I'm not too negative here. I just want to give my reality. And I will say that there are, I would have to say, numerous concerns out there where people who have done research and have not got the results. And then there's about an equal amount of people who have done this and have got great results that they think are very good. So I just want to put that reality, that's to me the reality where soil health testing is right now. But I want to end up my talk here with what I think maybe the most important soil health test is. And I'm going to say it's you and your shovel. So after all that, you going out there and visibly looking at your soil, counting earthworms, seeing the aggregate stability, what does your field look like as a cover crop or that doesn't? To me, that is the most important soil health test you can do. And yeah, you got to get a little educated on what you're looking for. I'll just give you a quick example. Last week on Saturday, we were racing to get done planted corn before the next rain was coming in. We had a 1/4 inch of rain overnight on Friday, and the soil was perfect before that. We had a 1/4 inch rain. More rain was moving in. We wanted to finish, and my son made a comment, "Wow, it's just so nice. We have these cover crops here and long-term no-till because planting was not an issue and the soil was just so nice." Yeah, it was damp, but I said, yeah, it looks like that chocolate cake that we talk about. So, and again, that's just an observation there. That's my soil health test observation. So you and your shovel is probably the most important soil health test out there. I would like to open it up now for some comments, and I'm sure some of you probably have experienced some very positive results. I'd like to hear about them. I'd like to hear about those who have not had the positive results. So I got everybody's mic on now. So who would have a comment or an observation, or you want to challenge me? You think I'm too negative on it? That's fine. I understand. Or whatever. So, who wants to comment on the soil health, or if you have a question about these soil health tests?
Well, I'll jump in then.
Go ahead, Scott.
Scott. Yeah. Well, to me, it's nice seeing some data. I'm definitely a data person, so it's nice to see the data and see you go through it, because I understand that with the variability when you, like, there are statistical measures for how variable your plots are, but when you look at those numbers, that's the thing. They're up and down, and then when you had the good soil test, or the good Solvita and the low yield and the low Solvita and the high yield, then you want to be careful with them.
Right.
Now, I guess one test that I see a lot on social media is the soil your undies test.
Yeah. I should have brought that in there. I never thought of that, Scott. That's good. That's a good one there.
That's the people, Steve.
I've never tried it or anything, but I see it as a social media thing. So, or it's a big thing to put out there. So.
Well, I gotta tell you, I that's great. I can't believe I forgot that. When I was in France speaking, they get this. Can you imagine this? They ceremoniously gave me a pair of underwear, and they took a picture of me holding everything just for the soil health, soil your undies movement. So, oh yeah, I got to try that too. I've never done it here in my own farm, but That's a good one. We should probably do a whole seminar on that sometime.
I
So,
could.
Steve,
Yeah, go ahead, Dan.
one thing's with the undies test. I'm all for, equality. So, I also do panties.
Well, okay. I'll take it a step further. That is actually what I was given in France, just to make it worse. And they took pictures of me, and I immediately texted my wife and said, Sherry, if you see a picture of me, I'm just telling you I was given this, and you need to know that. So I wanted her to know it before she heard it from somewhere else. So yes, point well taken, Dan.
Yes.
Gets people's attention. It sure does. Oh, I see Dan here. Dan Perkins says, I would second the management system's value of the PFLA test. It confirms to a farmer that they are going in the right direction. And I certainly agree with that. Greater education tool to a farmer wanting to dig deeper into soil organisms. Yeah, it's well said there, Dan.
And can I follow up on
Yeah,
that? I
go
mean,
ahead.
part of it is the PLFA is a very good test, but part of it is if we've been in a till system, I mean, we don't have the diversity. It's primarily bacteria that's in the
Right.
soil.
Well,
And
and that could prove a point to farmers, because sometimes for some farmers, that's all they need. For some farmers, they could care less. It's all about yield, nothing else. But farmers who care about the soil health, if they can actually see a test that compares that, that's going to make them think twice, and they may feel a little guilty when they're out there tilling the soil, if you know what I'm saying. And sometimes guilt is a good motivator. I'm not saying it is always, but yeah, but a good point. But Dan, while you're on here, you're working with a couple different things, aren't you? You want to tell us about? I wouldn't mind hearing your perspective on this and what's happening.
Yeah, I mean, I've worked quite a bit with all of these, and I guess inconsistency is the summary of it. So I've kind of trying to focus in on what are the really key ones, and one of them is the active organic matter portion.
Yeah.
It's a pretty good indicator, and from that you can look at a little bit of a stretch, but there is a correlation there to available nitrogen, aggregate stability, and microbial biomass, because it's all tied in together. Sure. And part of this on the, when you look at a field, and like in your plot, Steve, you're looking at 3 different nitrogen rates.
Yep.
Well, I mean, we don't know what kind of additional variability is out there.
Yeah.
And I mean, it's just the way it is. And a biological system, temperature and moisture are huge drivers.
Sure.
And so, like, if you're using the cover crops and you've got more moisture, then you've got more biological activity, and that's going to influence your CO2 burst and other things.
Yeah.
So what I'm trying to do with the active organic matter, I want to see just how robust it is. And I just started this spring.
Yep.
But I want to see if, for example, if a person is just starting out in cover crops.
Yeah.
1 year, 2 year.
Yeah.
Can we measure a difference?
Right. Yep. And that was my motivation when I first heard about them. Of course, at that time I was selling cover crop seeds, and I'm like, wow, if we can just show an instant result, this is going to be huge. And it's not quite there yet. So Dan is asking here, any comments on how often to pull these soil health tests? He asked Haney every year, PFLA every 4 years. My reaction is, it depends how much money you have. But I think if you want to monitor a field, there is a reality of cost here, because they're not cheap. So every year certainly would be ideal, but it's probably not practical, at least on a larger scale. So it's kind of the same answer on just the traditional soil test that we've taken. I've gone to every 3 years. That seems to be maybe an average that's out there. So anyway, I do want to get to some other people here, though. I want to hear from anybody who has used soil health tests, that has experience with them, in your comments. Is there anyone else on here that has actually used soil health tests and what their comments are?
Yeah, I've used them a couple of times and had pretty good success.
Okay. Tell us a little bit more about that, Cody.
It's just been on about a 10-acre field. About the last 5 years, kind of interesting. I've done replicated, I guess, samples, all grid sampled, and we had two sets of samples pulled, one soil health, one standard, and I've kind of kept track of them.
Sure.
Tried to do spring and fall, and I've actually followed the recommendations from the soil health test, once for spelt, and once for soybeans, and had really good success.
Good. Yep. For the rest of us, Cody's from North Central Ohio, just to get that straight. Well, that's good to hear, Cody. I know it's out there. I know there's success stories out there because I've heard about them.
The only wild card I've noticed is the organic matter levels in the soil health test compared to a standard soil health test. It's far different. Seems to be quite a bit more on the soil health test versus standard test, just in my limited experience.
Well, I know there's different ways to test organic matter, so there's probably an answer behind that somewhere on that parameter. Anyone else on here that I think maybe Bob was going to say something? Bob, have you done some soil health tests?
Yes, Steve, I made one set of it 2 years ago in my third year cover cropping, and I compared the cover crop compared to no cover crop.
Yeah.
And in just the 3 years, we could see a slight difference.
Okay.
That's the only experience we've had with it.
Which test was that?
Cornell.
I figured because you're from Western New York. Yeah, that's good. Now I'm in line here to do some pretty extensive testing with Cornell tests this year. As you may have expected, I've kind of pulled back on this testing a little bit, waiting for them to get more consistent and so forth. Like I said, I'm their biggest cheerleader to get it working right, but kind of excited about this coming year, working with the Cornell one just to see what that is. And honestly, I'm a farmer like a lot of you guys are, and even though I'm very, very interested in this, for me to spend the time and effort to do the Cornell test is a stretch. That's even for me, and the cost, which I'm not exactly sure what the actual cost is to a farmer. So, that's why I like to collaborate with people. They'll come in and do the work and I'll get the numbers from it. So yeah, I'm looking forward to seeing how things look on my farm this year, because I've been done very limited on any other tests other than the Haney tests. Dan?
Yeah, it's that Perkins has got up there that, it's like $120. And I was with the Soil Health Partnership there for about a year and a half, and we did a lot of the Cornell, and it was, again, these are fairly good soils for the most part, and they always, on the chemical side, they came out just super, but it kind of fell apart on the biological side. I mean, with cover crops, no cover crops. And part of it is, I think, there's a lot of fields that were new to cover crops, and that could have been part of it. But it again, saw very inconsistent results with it. I'd like to go back to that organic matter, and I think there's some things in there that, when you send in for a standard soil lab, they're doing the combustion, and the depth of your sample is critical. You got to be consistent. So if you shallow it up a little bit, that's a good way to increase your organic matter levels.
Yes.
You happen to get a little residue in the soil sample.
Yeah.
That can increase your organic matter levels. So again, this is part of working with the organic matter test. That I've the passive part is not going anywhere unless it's lost with erosion.
Right.
And it's the active portion is what we can see the changes.
So
what I want to do is couple would get Haney's new the CO2 burst where he has the computer chip on the lid, you put in the mason jar, and so I want to look at that active organic matter and the CO2 burst with the hooked to a laptop so you can see the chart
of
how the CO2 is changing.
Yep.
I've had some issues with that depending on your timing of you taking the sample and like vertical tillage tool, and we started with air, and it got much higher burst than the no-till. Well,
that makes sense.
But it was a short-term burst and then lose it versus slow and gradual.
Right. No, all good perspectives. Dan just said here too. By the way, Dan, thanks for the cost in the Cornell test, $100 to $120. I did not know it was that expensive. That's fairly limiting for most farmers, that's for sure. Dan also mentioned that there's different ways to check organic matter. So I'll just pass that along. You have to ask your lab to find out what they do. Other people that have done soil health tests that have experience with them want to give some comments or weigh in here.
I was just wondering, and this Don,
yes,
but I was wondering maybe the two Dans can pitch in a little bit too, but I've always heard the Cornell test was not correlated very well to Midwestern soils, and I'm not sure exactly why. So Dan said he was seeing some differences. I wondered is that because of the correlation to Indiana soils, or what's the situation with that?
Well, this Dan Towery, part of it is again Western New York. We have a lot of dairy farms and a lot of organic farms. These my understanding is these were heavily used to calibrate the Cornell test. And with the Soil Health Partnership, anyway, they decided that you're going to look at the calibration for the Midwest. And was it the soils difference? I mean, we've actually got, I very grossly say better soils in the Midwest than they do in Western New York. But again, we're looking at just the topsoil but is it the management? Is it the manure application on a regular basis? Is it the crop rotation that's used?
Right.
The cover crops that are used? Did that skew the results higher? And that we think that it is. Obviously, it's a combination of things. So, but
no, there's so many variables, yes. And the hope was that this would eliminate a lot of those variables. That was the hope. I would say that was my hope anyway, and I'm still hopeful. And I'm glad to hear that some people has worked out well. But also we're also hearing on this call here that there's inconsistency, but also there's places where it works. So I just want to mention here, you can ask any cover crop question you want. We could go beyond our topic, but next week I'm going to talk about cover cropping or double cropping after small grains. Now, some areas of the country this doesn't really apply. There's no opportunity for double crop, but a large part of the country it is. And typically you'll plant, cover crops or plant double crop soybeans after small grains. So I'm going to discuss the pros and cons and how to think about that maybe long term. So that's next week. And I'm just going to say for those of you who are regular here, I'm going to have to go to Thursday next week because I'm on jury duty. Now I hope I don't have to be in there very long, but if I'm there next Tuesday, I won't be able to do this show from the courthouse. So I'm going to do next week from on Thursday the 24th, though. Just want to let you know that. So other questions that you might have about this topic or any other cover crop question at all?
Steve, it's Wayne here. If
Yeah.
out of this group, if there was one test that everybody would agree on to use, year over year, what would it be?
Okay. Anyone want to weigh in on that? Which test is your favorite? Which one do you want? Which one do you like? Not sure if we're going to get a response to that, Wayne, or not, but anyone have a feeling on that? Which one are you most comfortable with? Or you've heard about most consistency? I'm not hearing any response, so I'm not sure. Yeah, go ahead, Dan.
I think this is the jury's still out.
Yeah.
It's again, you got to look at the cost. Are we asking too much? We know that the biology changes slowly.
Mm-hmm.
I want to be able to take like the soil petrometer, stick it in the soil, and it does all kinds of magic things, and it gives me 1 to 10. The needle moves if it's a healthy soil or not, and that's obviously being facetious, but it's it's because it's all relative. The soils are all different,
Yep.
and it's being how can we make it the best it can be?
Right.
But there are limitations.
Yeah.
So like the Cornell test, we talked about recalibrating. Well, we really need to take a look at by soil types, and maybe it needs to be calibrated. We lump soils, is it forested or prairie origin? Maybe those need to be separated.
Mm-hmm.
All kinds of northern part with the climate versus the southern part, like in Illinois, for example, huge differences.
Mm-hmm.
And so,
well, Dan,
someplace we need to draw some lines.
Dan Perkins has weighed in here in the chat. He's casting his vote for PFLA for biology tracking and for Haney for actual nutrient management each year. So thank you, Dan, for being brave enough to get out there on the line. So that's good. I just thought of something else here that some of you may have saw. There's an email came around that I got from the Soil Health Institute. They are asking for where there's long-term research sites available, testing cover crops versus none, or soil health practices versus not as many. They're asking for those sites available because they want to help essentially find soil health testing in those plots to see. Part of the idea is to get some more consistency. So I will mention that the Soil Health Institute is addressing this issue of soil health testing and trying to get it more consistent. So there is hope out there. I want to make sure that I'm abundantly clear with everybody here that I really believe that we're gonna move to more consistency on this to be able to use it more as a management tool. So that's just another item to add.
So Steve, one that I was investigating and didn't pursue it, but actually it was doing soil DNA analysis.
I've heard about that.
And it $1,000 a sample.
Okay.
And we know how complex the human DNA is.
Right.
Well, guess what, folks?
Mm-hmm.
The soil DNA is more complex than the human.
Wow. Well.
And so they can do all kinds of data, but
Yeah.
again, interpreting that we're years away.
Well, I think
okay.
you and all of us here that are in the cover crop promotion business have good job security. I don't think it's going to be all figured out very soon. So any other comments from anybody? I see Herb's on, Brad, Michael Brown, Avery, Amanda, anybody? Joy, I see Joy's on. You, any comments, Joy? Or anybody else? Questions?
No, I don't, Steve, but thank you.
Okay. I might see you on Friday. I'm going to stop by with that tour group that's coming through. So just.
Oh, yeah?
Wow. Yeah. Yeah.
Oh, I'm excited.
Yeah, it's good. Yep. Yeah. Tour group from Argentina. So.
Yes.
Brush up on your Spanish.
Will do.
Anyone else? Comments, questions, anything for our discussion today? Anything at all involved with cover crops? Well, thanks again for joining today. I really appreciate it. Again, next week it's going to be Thursday instead of Tuesday, and I'm looking forward to it. Appreciate your support and all, and thanks for joining with us today.